So this is a bit, I think, a work along. This is not really a lecture. I'm going through things kind of in some sense live coding. So as you'll see in this first session, there isn't actually much coding. But the idea is if you can work along, have the same RStudio up on your laptops, and work along as much as possible. In fact, I will occasionally just wait a few minutes, let you do something, and then kind of catch up with you. So the coding sessions or the modeling sessions are all meant to be very interactive. I won't be doing any more lectures from now on, unless in that last slot where we have the option to talk about whatever you want to talk. So this is, I assume every one of you uses RStudio. If someone of you decides to not use RStudio, then I assume you're advanced enough that you know what you're doing, and then you're on your own. You translate it to whatever system you're using. You don't have to use RStudio, but it's very user-friendly and it's commonly used. Most people use it these days, so I assume that's what you're using. So when you first started, you see something like this. Usually you have 4 panes. We don't have that right now, but I'll start it. Most often it looks like this. Up there in this top left, you write your code. Down there in the bottom left, it's the actual R. That's where you type in your commands. And then these are kind of sort of helper panels that you'll see in a second what we can do with them. And then we won't do too much to it, though. We'll get to it actually working through this, but we'll start out with the graphical interface. So the first thing you want to start for anything in R, you usually need to load some packages, which are these things that give you extra functionality. So in this case, the package we're using is that DSAIRM package. So when you start it up, it tells you what you can do to get the menu, and I'll do that in a second. But I'm actually going to just stay out of the menu for just a second. As I mentioned yesterday, I know that there's different levels of skill when it comes to coding here. So I hope for some of you that's walking through, starting with the graphical interface is just the right pace at which you need to go. For those of you who are pretty comfortable writing code, maybe that's too slow and you want to immediately jump to the more, closer to the code approach. So I'm going to sort of flip to that real quick, and then you can decide which way you're going to do it. You can either then follow along the way we're doing it now, or you can immediately go to the other option. So in general in R, when you want help on anything, you can do question mark and then the name of the function. It's usually a function or the thing you want help on. Now in this case, you can actually sort of ask for help on that package. Not every package has that, but I think the better ones have a little something where it tells you what it is. And the most important one that at the bottom, it's sort of auto-generated, is this index. So you can go here, and that gives you sort of all the functions that are in there. And the ones that are of most of interest to you as a user are all these functions that start with the simulate, because these have the actual models inside of them, and that you can run them. So for the first one that we're doing today, we're doing the acute virus ODE. So you can click on here, and that gives you the help file for this. If you're new to R, these help files are often a little bit hard to use. So the best thing often is to just jump to the bottom and look at the example. And so then you can run it and play with it. Once you get the hang of it, you understand how they work. So they always have a little description, then they show you how you use it and what sort of inputs the model wants. In our case, the models, you always need starting values for the things you track, and then the parameter values, and then for how long you want to run it. And so this is all described here, what you give into it. And then, there's some more details here on what you get out of it. And so you could, if you wanted to, run each one of those models just directly. So, we could, for instance, do this here, as it shows in the help file. This runs this model with just the default settings. And so if you might have seen, if you do it on your computer too, something shows up there. So that means basically the results from the simulation run are now stored in this object that's called result. You can look at those objects. That might not tell you too much if you're new to this, but the idea is that you have everything here in what's called a data frame, and a data frame is basically just a thing in R where you have a bunch of columns and a bunch of rows. And most of the time what we have is every column is one of the variables we track. So in this case we have uninfected cells, infected cells, virus. We have some innate response, which is called F here, and some adaptive response, which is called T here. And they're all tracked as a function of time. So you have a model run, you start at some time 0, that's usually the time you start, and then you run the model and you track how each one of those changes over time. And that's basically the information that is stored in there. And most of these apps, at least the basic ones, give you something back of that type. And then you can play with it. You can, for instance, here is a way to plot the U variable, the uninfected cells, as a function of time. So you would get a plot like this that you see in the top right here. So you can interact with all these directly, and that's what I call level 2. And we'll do that in the next session, the next modeling session. For now, I'm going to switch to the graphical interface way of doing it. But if you feel like you want to directly go this route, you can go ahead and do so. And basically just call the R functions with the settings. So, here in the help file, you see that you can change things instead of using the defaults. You can set things to different values, and then you would get a slightly different answer out. In this case, I don't think it looks that much different, maybe a little bit. So that's just for some of you that might be interested in doing that. And then the level 3 one would be, you can actually pull out that function and look under the hood into the function. So the easiest way to do it is you load the package, and then the menu. And then here at the bottom, it's the download R code for all simulations. And that's just a zip file that you can download to your computer. And then in that, there's the actual code for each one of them. And then you can basically look into the code. And of course, if you want to, you can modify the actual underlying R code. We'll play a little bit with that tomorrow. But for today, we'll stick first with the graphical one and then sort of the way of calling these functions, but we're treating the functions for now as a given. Of course, this is for learning. It's very unlikely if you start doing your own research that one of these pre-written functions do exactly what you need them to do. What you would then need to do is you maybe take one of them that's as close as possible to what you want to do, and then take the code and modify it to be suitable for your specific research project. So really, this package is mostly meant for learning, for teaching, because it has these pre-written functions, but they're there for you that if you want to adjust them to your purpose, you can do so. Okay, and as always, if you have questions, I have the chat window open. You can type it in there, or you can just unmute yourself and ask it if anything is unclear as we're going along. So then, since Paul talked about acute viruses earlier, this is what we're now playing with. And this is this one here, the acute virus and immune response model. If you played it with it, you might have seen in the basic virus one, it's actually so basic, it doesn't even have an immune response in it. There's just the uninfected cells in this model, uninfected cells, infected cells, and virus. And with that model, if you played with it and ran it, you can see you can already get something that looks like an acute infection. You get the virus come in, it grows, and then it runs out of cells it can infect, and then it goes down. For those of you that are familiar with sort of a population level model, very similar to an SIR model where you have sort of a pathogen like SARS or something, or flu sweep through the population, cause an outbreak, and then die down. So it can happen without the immune response, but we do know that the immune response usually plays a role in affecting the dynamics of the infection. So we're looking at the one here, the acute virus and the immune response. And so each of these apps is kind of structured in the same way. You have your main sort of screen here on the left, you have all the inputs into the model. So in the inputs are always the starting values of the different things you track, and then the parameters. And the parameters are really the most important ones. They tell you, how quickly do they grow, how fast is the decay rate, at what rate does killing happen. Now here for this kind of teaching, I'm just setting some values that give somewhat, outcomes that look kind of reasonable. Now for any real research, of course, you would like to go to the literature and try to see if you can find values from the literature that are as close as you can get. And what you'll find quite often that it's really hard to find those values. Because a lot of the published literature is still very qualitative, but no one's trying to really measure the rate at which T cells kill infected cells or things like that. So quite often it's tricky to get those numbers, but you should try your best as good as you can go. And then those is what you put into your model if you want to simulate something specific like flu or RSV. So, you might even use a very similar model for the two pathogens, but you might adjust the parameter values based on the details of the biological system. So these are your inputs here on that slide. And then when you run a simulation, you get basically a plot like I just showed you on the console, just a little bit nicer looking, that tracks all the quantities that you're running in your model. So in this case, again, uninfected cells, infected cells, and virus. And then we have an innate and an adaptive response. And it also gives you, the diagram is nice. You can see the whole time course, but sometimes, it's hard to read off certain numeric values. So as a bit of a help, it also spits out some of the numerics, kind of the min, the max, and the final value. For most of them, that's what you get here. You can also, if you haven't played on, you can change the plotting. The ggplot is sort of the default, which is used very often to make very nice plots in R. Plotly is nice because it's interactive. So if you make a plotly plot, you see it looks slightly different. But the cool thing is that, you can do things like, you can zoom in. And, then you can auto scale, or you can zoom in like this, and it lets, or you can click on one of those here and then it will turn them on or off. So let's say I only wanted to see the uninfected cells, I can turn those off. So that's a nice thing about Plotly. Of course, it's interactive only on a website. So as soon as you put it into a manuscript or something like that, that doesn't work anymore. But if you have something like an HTML document, or you put it on a web page, this is kind of a nice thing. So here in this one, you can use both ways of doing it. And then underneath that is what arguably as important as what you can do here, the whole explanation of what you should do. So first, there's always a brief overview that just tells you what's going on here very briefly, and what hopefully you learn when you do this. And then the model is one of the important ones. As I mentioned yesterday, all of them have sort of the same structure. First, it's in words describe what's going on, which compartments or quantities are tracked and what processes are happening. After that, you see the diagram, and then you see the equations of it. So when you build your own model, at some point, this is the logic way you go about it. You decide, okay, which quantities do I want to track based on the biological system and the questions I have? So then you decide, okay, what processes do I want to track? So you start with this sort of verbal one. And then you might want to scribble it down as a diagram. Once you get really good at it and have lots of experience, you can go straight to the equations. But for beginners, it's always good to do this. And I think in general, this is just helpful to scribble down a diagram. You can just do that with pencil and paper even. And you say, okay, these uninfected cells, they get infected by virus at some rate. And then these infected cells, they produce virus, and the infected cells induce the innate response. And so, you just draw these diagrams and put arrows from which one is going where and who and which thing affects what thing. And as you can see, even though we only have 5 things here and only track very generically just an innate response and an adaptive response, there's already a bunch of arrows and things interacting with each other. So these models can get fairly complex fairly quickly. So that's why I generally recommend start simple. And then once you've explored your simple model and you realize it doesn't quite yet do what you need it to do, put in more details. I prefer that approach over an approach that tries to put everything into the model, because then it often gets so complex that it's really hard to understand what's going on in the model. And then in the end, at some point you have to translate it into a version that the computer can understand. And a very common one is to do differential equations. So again, on the left side is just the quantities you track, and the dot is change with time. And on the right side is all the different processes or mechanisms that you want to model. So here, this is the rate at which uninfected cells meet virus and get infected at rate B. So this is basically from here going into the I compartment. You see minus here, that means an outflow, a reduction of this, and sort of a plus sign here, and you see it arrives here. And the infected cells die at a rate, DI, so that's outflow at that rate. So that's how you build those models. And so for every app, they're described. There's a hand raised. Oh, okay. Go ahead, ask.
Yeah, can you just real briefly go over what's the difference between the dotted lines and the solid lines?
Yeah, so what I'm trying to do, and unfortunately when you go through the literature, that's not very systematic, but I find it useful to have solid lines that mean actual flow of entities. So, you know, this is a real uninfected cell, an actual cell that becomes an infected cells. Or here, this one, this is an actual real infected cells that gets killed by the adaptive response and dies. Versus the dashed lines, I indicate interactions. So the infected cells don't turn into innate response, but they induce the innate response, but it doesn't change the number of infected cells. So that's what I'm trying to indicate with the difference between solid and dashed. One is actual, what I call physical flows. So, you know, things go from one state to another, versus the other one is just one quantity affects another quantity without itself changing. I think it's useful for teaching. Unfortunately, again, if you go through the literature and you see these diagrams, people don't always do that. So sometimes people just use solid flows. Sometimes they use colors. So, you know, you can find different ways. But here it's just kind of trying to help you distinguish between physical flows and interactions.
Gotcha. Thanks.
Okay, so this describes the model. After you read this, hopefully you'll get kind of an understanding of what's going on. And then sort of the bulk of really what's actually going into these apps, the writing the code is actually not that hard. At least once you've got a lot of experience with it. But then all these tasks. So the idea is that as you work through these tasks, you will learn whatever I thought would be good to learn with this specific app. And so, you know, you can go through it and then, you know, just follow the instructions and do that. And again, now in this case, what we're doing is we're just doing everything in that graphical interface. So we're setting them here and running them. However, if you felt like it, you could also do it in this framework here that we looked at earlier. So, you know, you change your initial settings here and then run it for different values. So you could either more directly interact with the code and skip the graphical interface, or you could go through the graphical interface. And so, you know, the recording here is not so crucial right now. It is, you know, if there was a situation like a course where I needed to assign grades to students, I would ask them to record these things and submit it, and then it would be graded to see if they got it right. But, you know, for you, it's just, you know, you can record it, you don't have to. But so the idea is you work your way through the tasks, and hopefully by the end of having worked through the tasks, you will have learned whatever the topic is that I'm trying to teach you. So that's what, you know, you probably spend, if you go through these apps, most of your time on this one here. And then further information is just always tells you what the underlying function is. So in this case, we've already looked at this, the simulate acute virus ODE, and remind you that you can either use help or the question mark that I showed you to pull up the help file for it. And then sometimes there's also additional reading where I say, okay, if you want to see similar models like this in the published literature, you can look at these papers here. So sometimes those exist, sometimes certain apps don't have them. But so this is basically just if you want to go further, it's down there. Okay, well, before we dive into actually going through those tasks, any general questions or comments on it? Okay, I interpret the silence as meaning it's all clear for now. So then let's jump in. Let's start with the first task, and I'm actually going to give you a minute or so you can go ahead, read through it, and actually try it yourself. And then I'll jump in and redo it here, sort of in front of you, so you can compare what you did with what I'm doing. Okay, I'll go ahead now and start talking through it. If you're still working on it, that's okay. You can just keep going. We might end up at the same time at the outcome. So, you know, basically this tells you how you should change all the settings over here to get what you need to get for every task. And, you know, then hopefully the results will match. So the 10 to the 5 uninfected cells, no infected cells, and 10 virus particles, that's actually already preset. So sometimes the defaults already match what's given in the first task. Sometimes I'd purposefully make them different. But in this case, those three are matched. So it says here, absence of immune response, and it comes later here, variables F and T and 0, right? So if we have no immune response, we don't want to have any to start out with. So we're setting those two to 0. So then next one, infected cells have an average lifespan of 1 per day. That's a thing that, if you're new to modeling, it's something that will likely trip you up a few times, so you have to used to it, that in the models, it's almost always, it's rates. The rates at which cells die, or the rates at which new virus is produced. Well, it's easier to think about it, and it's often, if you find it in the literature, it's often reported in the inverse of the rate, which is sort of the lifespan. So, in this case, if the lifespan is a day, then the inverse would be the rate, 1 over day. And then you also need to always be careful about the units, right? Your model runs in a certain kind of units. That's up to you, the units you want to let it run in. But once you've decided on your units, and all your parameters and everything needs to be in those units. You can't have a parameter one that's expressed in days and another one in hours, and that will just give you the wrong results. So you decide what you want to run the model in. Usually there's a natural unit. If we have an acute infection like that, we might either use hours or days. If you had a chronic infection, maybe your units is actually weeks or months. You decide what the most natural unit is for what you want to run it. In some sense, it doesn't matter for these ODE models. They're somewhat invariant. So you just, when it comes to the time unit, as long as you always have your parameters in the right units. So in this case, the first one is easy. If the lifespan is 1 over day, then the inverse of that is also 1. So the rate at which cells die, DI, is 1. We've already have that in there, so we don't have to change it. And now virus is a lifespan of 6 hours. And again, we need to take the inverse. But first we can convert it 6 hours, or we can convert it 6 hours, that's a fourth of a day. Right? A day is 24 hours. So we need to go into units of days. So we have a fourth of a day, that's the lifespan of a virus, and then we have to convert it to a rate, so the inverse would be 4. So instead of the 2 that's already preset here, we need to change that to a 4. So that's something you need to always think about carefully, and when you come to these rates, you make sure you do that right. That is a very common thing where someone just types in 6 instead of doing the conversion, and then the results aren't right. So that's something always worth checking if what you're getting is not matching with what you think you should be getting. So virus production to 100 variants per day, that's here, we have that. The rate at which cells become infected, we already have that in there too, so those are good. No need for unit conversion, so that's this G parameter. I think in the basic virus app, I discuss what that means and how it works. So here, we'll just ignore it for now. We'll just set it to 1. And then we don't want an immune response, right? So we already set F and T to 0, so we don't start out with 1, but then we also don't want any production of any immune response. So we said, this one rate of induction to 0. And the rate of decay doesn't matter, because if there's nothing there, nothing can decay. So it doesn't really matter if we set this to 0 or 1 or whatever. So you can just leave it at this. It doesn't matter, because you have that outflow out of that F compartment. Well, if there's nothing in there, nothing can leave, no matter what you set the rate to. So it doesn't really matter. And again, rate of innate immune response action doesn't really matter, because if there's no F around, it doesn't matter how strong it acts. Nothing happens because it's not there. So again, you can leave that at whatever you want. You can set it to 0 if you're more comfortable with setting them all to 0, but I'll just leave it now. Okay, here, rate of adaptive response induction. Again, we don't want any induction, we don't want any production, so we set it to 0. Again, here, if there's no adaptive response there, it doesn't matter if we have a decay rate for it or not. And also the strength of the response action doesn't matter. Simulation time, almost always we start with 0. It really doesn't matter for these kind of models. So, you could start at 100 and you would get exactly the same shape. The only thing would happen on the x-axis, instead of the first number being 0, the first number would be 100. And so it's very common that you always start at 0. And 0 is kind of the time of infection, or whatever you think this is a natural 0. T final is just for how long you want to run the simulation out. In this case, it doesn't matter too much. Sometimes you will see, we'll get to that when we do the chronic one, where things wiggle around until they settle down, and then you might need to run your simulation long enough for everything to have settled down, if that's what you're interested in. Time step is also for these ODE models, it doesn't matter. If you have a discrete time model, which some of the apps have, that you can explore, then it matters. You usually want a small enough time step such that it doesn't make too big a jump. For these kind of models, the time step is really only relevant for where it returns it for your plotting. So you want a time step small enough that you see kind of nice results. The trade-off with the small time step is the smaller the time step, the longer it takes the simulation to run. Now, in this case, they run really fast, so we don't really have to worry about it. But if you have a more complex one, sometimes you might want to not get it that finely spaced. Again, this is not the time step at which the underlying numerical routine runs. This is just the time step for where you want results for your plot. So that should do it. So let's see if we run it, if I did everything right. Final number, yeah. So we basically got what we wanted to see here. You can play around here, and you can, and also always reality check, right? We don't want to see any F or any T here. We only see U, I, and V going up. If we saw any F or T going up here, we know something wasn't right here the way we set it. And then we have to check again. So always make sure that what you get here corresponds to what you expect to get. Cassini, you can switch. Usually, almost never for time does it make sense to plot it on the x-axis. Very rarely. So most of the time you leave that on a linear scale, but sometimes the y-axis, you can get such large numbers that it might be worth to plot it on a log scale so you can see it better. So if we did that now, and we can also switch back to ggplot if we want to. Looks different now. So you see nothing here. You see, you can't plot 0 on a log scale because then it's minus infinity. So what actually ends up under the hood is that it adds a tiny little number to this such that you can show it in the plot, but you see basically there was 0, the innate and the adaptive response. And you can see the sort of standard pattern. Virus comes up, and of course infected cells increase at the same time, then the uninfected cells drop down, and at some point there are enough of them gone that it can't sustain itself anymore, and then you get the decay. Okay, that was the first task. I'll stop here. Any questions on that?
Yes.
Okay.
Actually, just one comment. The difference between setting all the immune responses to 0 compared to leaving as it is, in terms of like the strength of the innate immune responses. And it doesn't matter the number because there is no starting point in the first place. The difference between setting up to all 0 is that those lines are not going to be included in the plot for the immune response. So we don't even have F and T plotted there at all, which is cool. For imagine if I plot this in a paper that.
If I set them all to 0 here?
Yeah, if you set all to 0, the green and yellow line.
They stayed for me. I just ran the simulation again. It could also be if you use Plotly versus ggplot.
I did ggplot, yes.
And then if you do a log or not log.
Yes.
So, but here they're there. Okay, now here they're gone. So yeah. Basically, it just is a function of the plotting where it cuts it off or not, right? So that's kind of the only difference here. In either case, they're basically 0 and stay at 0. So yeah, it might just be the way that when you make that plot, it shows. Yeah, you definitely want to decide that in a paper you might or might not want to plot them, right? You might want to show that they're present in your model, but they're actually 0. Then you might want to include them, or you might want to say, well, since they're not playing any role right now, I don't even want to plot them. So that's, I think either way is justified. Depends on what you want to show.
Mhm.
And you said you had another one?
I did. The question was about the infected cells, right? So we would go to I, and it's 1.5 E06.
Sorry, say that again.
No, record the number of infected cells at the peak of infection.
Yeah, so this would be here. Maximum, sorry, maximum
Okay.
maximum value of I. So that's what you would record.
Got it.
So there are two ways. Usually I make it such that whatever I ask people to record, you can read off here, just it actually spits out the number. So all you have to do is round it. You could, again, as I showed earlier, a nice thing, I guess I didn't actually fully show that. A nice thing with Plotly is that you can also, as you move over there, you see it shows the numbers, the actual numbers underneath. So you could, kind of try to sort of zoom in here into this part, which is kind of the peak, and then move over here and, try to read it off, right? So you see 18,305. So I guess we actually found the peak here. It's not quite as accurate as down there, but, if you wanted to read it off at, let's say 15 days, then this is the way you could do it with Plotly.
And I have a third question. Sorry.
Sure,
Is
that's
there
fine.
is there a way to extract the raw values from these steps from the package? Let's say a collaborator asks, look, but I want to know, it makes sense. I was thinking about a collaborator asking, okay, the final time you set up was for 50, but I want to know exactly at day 15 what is going on over there. And we can extract from a table or...
Yeah, so that's this thing here that I briefly mentioned earlier. This thing that the simulation function gives you back is this data frame that, for each time, it basically has every variable as a column, and the row is each time step. So you would have, you have time 0, 0.1, 0.2, and so on. So you would basically go down, you would find time number 15, and then you would go across and you find U or I or whatever you want, and there's the number in there. So yes,
cool.
with this one, if you run it this way, you can pull it all out, and we'll play with that later today as well, how to do this from here. Okay, there's a question in the chat. Let me see. Okay, it's a bit technical. I'll try to give a bit background. So when you have these compartmental models, like here, we are assuming that uninfected cells move into infected cells, and then after some time, the infected cells die, and they basically move into a class of dead cells. Here, we decided to not actually model the dead cells because they're not playing any role in the system, but one could model them if one wanted to. In fact, you might remember that in the model that Paul had, the dead cells were actually in there, that we built for flu. The reason why we had it there, because there was some indication that in vitro system, the dead cells might actually regrow. I mean, not the cells itself, but the dead cells might be replaced and new ones might be growing in their spot. And so that's why we included it here. In this case, I assume once the cells have died, they don't play any further role, so they're excluded here. But so basically what happens here, the cell moves from U to I, then to the unmodeled D compartment. And so the way this is modeled with this, rate DI, I, what it assumes underneath with this kind of model is that the average time a cell spends in the I compartment is the inverse of DI. But at any time, there's a chance that a cell moves from I compartment to the next compartment, in this case, the dead compartment. Basically, it simulates sort of a radioactive decay. It's an exponential process. You have a bucket of radioactive material, and it takes about 100 years for half of it to be gone. But any one of these atoms could decay at any time. Some of them will decay within seconds, and some will be around after 500 years. And so that's kind of the same assumption you make in these biological models, that on average, the infected cells live for a day, but sort of any individual infected cell could be gone within a second, or it could be hanging around for 10 years. Now, that's, of course, not realistic biologically, right? That's an approximation. It's sometimes okay, but not always. And so what you might want to do at some point is make that a bit more realistic by saying, okay, the infected cells are infected for at least, let's say, 5 hours before they start dying. And then after, 50 hours, all of them are dead, right? No one lives beyond 50 hours. So if you want to have something that's a bit more realistically distributed, then these very simple ODE models like we have here, like this one, doesn't work anymore with just this single one. A nice trick is what one can do is, and you might see that sometimes done, is with what's called often dummy compartments. So instead of just having one equation for I, you would have this I equation like more or less replicated a few times. You would have I1, I2, I3, I4. And so what happens is that initially the uninfected cells get infected, they move into the I1 compartment, and then they sort of snake their way through from I1 to I2 to I3 to I4, and only once they reach I4 can they die. And what that trick gives you is that then the overall time the cells spend in sort of all the I compartments together becomes more sort of a gamma distribution. So it basically doesn't have this exponential one, but now most of them are infected for a certain time, and then after a fixed time they go and leave. So that's a way you can do it, and that's done both on the as the question suggests, at the SIR population level and on the within host level. People often do that too. So yeah, so the same tricks you can apply to modeling on the population level, you can absolutely use here, and people use more or less the same models. There are other ways to do it. You can use delay differential equations. You could use partial differential equations to achieve the same results. The problem with those is that they're just harder to work with numerically. So I would generally advise to only go to those if you really have to. Usually these dummy compartments work pretty well. As soon as you have like 4 or 5 of these dummy compartments, it's already pretty much almost like a step where almost everyone is this long in the compartment and then they all leave. So that's a good way of approaching. I hope that answered the question. If not, please feel free to follow up. Any other questions? If not, then we'll just move on. And again, let's, you've got the hang of it. Let's do task 2. I'll give you a minute or 2 to do it. And then I'll redo it here. If you've done it, what do you find? Okay, one person, David, said it's the same. Another, it's the same. Has anyone worked through the basic virus app or otherwise knows why it is or should be the same? Okay, one answer is it depends on the starting value of U. That's part of it. U does play into it. But we changed a whole bunch. We left U the same, but we changed a whole bunch of other stuff. Why did it not change?
So I think if you look at the model, if you're doubling the virus production and the infection rate, but halving the lifespan of the cells in the virus. So if you double virus production, but half the lifespan of an infected cell, you're going to come up with, the same virus production. And if you double the infection rate and half the lifespan of the virus, then you're going to get, the same amount of virus present at any given time. If that makes sense.
Mm-hmm. Yeah, I think that's a very good intuitive explanation. So, you can just sort of logically think yourself through. You don't even have to know much, sort of formal, this reproductive number mentioning there. But yeah, this is, the rate at which virus is produced. So, right now it's 100 and then, per day. And so if the virus lives right now a fourth of a day, so it would produce about 25. So we know that make 200 a day, but we make the virus live only half as long. The infected cell only half as long. Sorry, earlier it was 100 a day. So now it's 200 a day, but it only lives for half a day. So it's still a total of 100 variants produced on average by an infected cell. And the same reasoning here. The virus has a certain rate at which it can infect cells. And of course, there's a competition between the virus being able to infect a cell before it gets cleared out of the system. So, if the virus is twice as good as infecting cells, again, the rate at which it infects cells is twice as high, but it also gets cleared twice as fast. Then again, this equals each other. So the same number of virus particles are able to go on and infect other cells as before. So basically, they kind of cancel each other. And so, then we should get the same. Now, you might have noticed that while the final result is the same, the kinetics looks different, right? So you don't get the same shape anymore. And that's an important one for those that have heard about the reproductive number. And in the day of COVID, most people have actually heard about it now on the population level. It's kind of a measure of transmissibility of a pathogen. In the population level, how well does it spread from person to person? But we can define very much the same quantity here on the within host level for the virus, sort of how transmissible, so to speak, is the virus to go, from cell to cell. What's important about the reproductive number, both on the within host level and population level, it doesn't have time units. It doesn't tell you anything about the speed. A good example on the population level is something like HIV and smallpox. They have roughly the same reproductive number, but smallpox is a acute infection that you get, then you're cured. HIV takes much longer. So the HIV epidemic, you don't measure in days or weeks, you measure in years. Because R naught only tells you the reproductive number, how many new, in this case, infected people, or in this case, on the within host, infected cells are generated for each starter, in this case, each infected person or each infected cell. But it doesn't tell you anything about the speed. But I mean, for any real outbreak, you need, both are important, the speed and how big it might get eventually. So here, the final result is the same, but the speed differs. And so in the basic virus app, that concept of the reproductive number is explored and discussed a bit more. And again, this is a concept that comes from, epidemiological modeling where it was first introduced, but a lot of that, because it's very much the same models, it also applies to the within host level. So, initially most of it went epi, it was introduced first, a population level model, and then within host modeling is more recent. So then people adopted that. I think at this point, it's actually kind of a back and forth, and there's some ideas and concepts that people apply on one level and then they use it for the other level and vice versa. Though still, especially now with COVID and even before, the population level modeling field is definitely bigger. There are more people doing that still than on the individual within host modeling. Okay, yeah, this was just, a basically recap. If you've done the basic virus app, it's basically just a revisit of this one, because we're basically running that model because we have F and T turned off. We're really only having U and I and V, which is exactly what we had in the basic virus app. So we're just, rerunning that here. Any other questions on this before we move to the next task? Okay, and then let's finally let the immune response play a role here. So start with it, and I'll come in a minute or so and catch up. Okay, Holly, I'll get to your question in a second. So I made the changes here, set these things back to the starting values here, and then I just turned RT on, DT, KT. So, there's no adaptive response, and it's in, it has a rate at which it gets induced. So, one could think maybe there's something going on, but, if we run the simulation, we see that no, there isn't. And then, the question is why not? And the reason is that the way we set up the model is we're assuming that the innate response is necessary to induce the adaptive response. So if we go back to our model here, we see here that the adaptive response is induced at that rate R, proportional to how much there is of the adaptive response and the innate response. So if the innate response is 0, F, then it doesn't matter how big the rate of induction is. There is no induction. So there's no adaptive response. That's an assumption we made, right? We decided that, we think the underlying biological system we're trying to scrape is such that you can only have an adaptive response if the innate response has been triggered, and the induction of the adaptive response is proportional to the innate response. That's an assumption based on science, right? Someone could come along and say, no, I don't believe that. I think my system is different. It needs to look differently. For instance, and we'll get maybe in this a bit, so they could have said it should be with the square root of the innate response. Or there should always be a bit of an induction, even if there's no innate response. That's not technically a modeling question. That is, again, there you need to know your science enough that you can say, based on what I know about this system, this is what makes the most sense. Sometimes you're genuinely not sure, and then what often is worth doing is explore a few alternatives. As Paul presented earlier for that influenza example, we built a few different models that all we thought were realistic or reasonable, and then try to compare them with the data. So you can do that. You can build a few models, and either with or without data. Even if you don't have data, you could run the models and see how they differ in what they're predicting. And then you can say, oh, based on what I know about the system, this one seems to be the more reasonable one compared to the other one. So there's very often this iterative thing going on here. But so here in this case, we don't get any adaptive response because there is no innate yet. Okay, now to Holly's question. If you could say more specifically which parameters, is it that rate versus lifespan part that's confusing, or which are the confusing parts? Okay, we'll wait for her to find an example. I'm trying, so in these apps, what I've been doing is I've been trying to find a balance between, just telling you, like, here I did RT equals 0 and DT and KT equals, so, you immediately know which one to go to and set it. That's of course easy, but that also doesn't induce a lot of learning. Now, if I do it more verbally and you have to figure out which one of that is, that might be at times a bit harder, but hopefully you will also be able to better start translating the words, the biology, the science into the models back and forth. So sometimes I might go too much in one direction, and sometimes I might have it not well in the other one. But the good thing is once you get going at that, you get a hang of it, and these models all look fairly similar. And I'm trying to be very consistent in my using the same letters for the same parameters. Unfortunately, when you go through the published literature, that's not necessarily the case. In mine, I always call the uninfected cells U and the infected cells I and the virus V, for instance. But if you go through the literature, you can find people, a common one is uninfected cells are called T, for target cells often. Infected cells are called T star for infected target cells, and virus might also be called V or so, but sometimes people call it X, Y, and Z. So unfortunately, when you go through the literature, there is no sort of standard way of calling things. Somewhat less so than you would see if you looked at the population level modeling. That makes it sometimes harder to read the papers. What I always recommend is look at it, take a deep breath, look again at the equations and see if what they're showing, even though it might look strange, is actually a model it's just they use different letters for things. The flip side is also true. Just because you always use the letter B for the rate at which cells become infected, someone else might use the letter B for the rate at which infected cells produce virus. So don't just assume because you see that letter, it means the thing that you're used to it. Some people define it. Usually in a good paper, there would be a table that gives you every parameter and its meaning and the values they choose and where they got these values from. So in a good modeling paper, there should be a table like that. Okay, yeah, the lifespan and the rate, these are definitely the ones that are initially the trickiest. That's something one has to get used to, that the more intuitive biological thinking is at lifespan, how long does the cell live, how long does the virus live, but these models always use rates. And so that is a constant thinking about it and making sure, okay, I need to translate it, out of the literature into my model. I probably have to transfer from lifespan to rate. And then maybe when I report it, I report it back at lifespan. Another quick caveat I want to mention here, sometimes you see half-life. And half-life has an extra factor of log 2 in it. So, lifespan and half-life are basically the same number, but, scaled by a factor of log 2. So that's also something to be cognizant about.
I think another factor of that may be that, if you have a hard immunology background, clearance is active. It's T cells clearing a virus or antibodies clearing. When here it's just from the system it's being cleared, either through just naturally just falling apart in this case because there is no adaptive immune system or anything like that that's actively clearing it. So maybe there's a little confusion there. That's how I think about it, at least.
Yeah, that's true. Again, terminology, it's for infected cells, it kind of is it makes sense to say that's the rate at which they die. But then again, even there, if you look in the model, the infected cells here, they die at that rate DI in some sense naturally, whatever that means. They die because they're infected or something, but then they have an extra rate of being removed because of the adaptive response, the T cells, right? So there's two ways they can die in two separate processes. And so what words you use, that can be, it could be death, it could be clearance, it could be removal. And again, there's no consistent way that people are using those words. So you just always, then at the end, there's no way around it, looking at the model, either the diagram or the equations, because only at that step does it get precise. And that's often a challenge, when you go from a question and you have a, question is I want to model, pathology. Well, as a model, I come along, well, define pathology for me. I need a measurable quantity that I can actually translate into a model. And then, often if you talk to an experimentalist, they actually get stumped and I don't know, how bad it is. It's okay, how do you measure it? And so you, for better or for worse, once you start modeling, you have to nail yourself down to something that is quantifiable and measurable. And people can argue about that, right? If you want to quantify, let's say, morbidity for flu. Of course, if someone dies, that's an obvious event. But if they just feel bad, what does that map to? You could maybe say, well, we know the innate response and interference are responsible for feeling bad. So maybe we map it onto the total amount of innate response. But someone else could say, no, the total amount is not important. The peak is important. So you could map it on that. And that's where a lot of the modeling, this, you have to make decisions based on what you think is most reasonable about the system. But yeah, definitely terminology, here for the virus, do viruses even die? Is it death? Is it clearance? Is it removal? In this case, it's all of this, them kind of leaving the system here at that rate. But you can also see they can leave this way. They can also leave this way. Mainly the virus goes back into the uninfected cells. That also removes the virus from the system because this is the free virus, technically what we're modeling here. So if the virus disappears back into an uninfected cells to turn it into infected cells, it's also removed out of this compartment. So yeah, it's definitely something that, initially one has to go fairly slow and careful to really think about these. After some time, once you've done it often enough, then, things will go faster. Any other questions? Okay, I think we might have time to do one more. We might not get all the way down, but that's fine. So I'll give you another minute, start task 4, and then I'll do it. Oh, also one thing that if you haven't found that yet, sometimes it's annoying to keep scrolling up and down. So at some point we introduce this thing where you can float your task list. So you can actually have this thing up here next to your inputs. So that might make it easier. It's just a convenience that you can gold stars for anyone that knows enough R coding to tell me how to get this thing above this bar. There are some things where I haven't been able. The same if you talk a bit on this here, you see the none and the plotly are sitting on top of the bar. They should be underneath, but I have not been able to figure out how to do that. Okay, so I made the changes. I'm going to run the simulation. And you see two things, that the innate response actually comes up. And you see another thing, that the virus doesn't change. And the reason for that is because we've, turned on the induction of the innate response and also its decay. We didn't let it act yet. We assumed its impact is not there. So it basically just comes up and goes down, but it doesn't interact with the rest of the system. It's often good when you build a model and you test it out, you turn on every bit by itself and make sure that bit behaves the way you want it to behave before you turn on everything. Because especially when the models get complex, if you have everything going at the same time, you might not even know if all the components work right or not. It could be that some parts don't actually work right, but they cancel each other out. And so the result looks okay, but you don't realize that it just looks okay because two wrong things happen to cancel each other. So, it's always a good idea to do it like we're doing it here. You start with the basic model, just turn on some components, make sure it behaves like it should. Then you turn on another component, make sure it behaves like it should. So quite often setting certain things to 0 initially and making sure what you expect to get is what you do get is a very good thing when you build your first model and try to debug it and test it and make sure everything works as it should. But of course, then eventually we want the immune response to do something. So what did I say? 10 to the -5. So now we let it actually act. And now what we would expect is that something happens to the virus. Let's see. Yep, sure enough, things change. So now the innate is triggered and the innate acts on the system. And, if we want to look what exactly the innate does, oops, that wasn't good. We can go back to our model here. So the innate response reduces the rate at which virus is produced. Usually it's reduced by P times I. And so now if there's an F there, we divide by 1. Usually we just divide by 1, so it's just P times I. But if there is some F around, then we divide by this number, which is larger than 1. That basically reduces this P times I, which basically means the infected cells now produce less virus. So the idea is that the innate response is some kind of interferon response that basically makes the infected cells somewhat resistant to virus production, and therefore they produce less virus. So that's what we're doing here in the model. And then as virus produced, gives us a smaller peak, as we do see here.
Could you repeat again, what does the strength to adaptive response have? Wait a second, that was not what we changed. The strength of the the innate response. What does that mean biologically? What is that? An example.
Yeah, so it basically means for every, whatever measurement unit your interferon is, let's say for every nanogram of interferon or whatever unit you have, if we're assuming the innate response is nanogram, this is how strongly it reduces the production of virus. So it's this Kf here in this diagram here. So, if you have F here, the innate response, but Kf is 0, that basically means no matter how much of that innate response you have, it has no effect. As Kf gets larger, it means for every whatever unit your F is, nanogram, milligram, some molar unit, whatever unit you measure it has a stronger and stronger impact on the reduction. Now, a parameter like this and similar ones tends to be one that are basically never biologically measured. So you will probably not find anyone in the literature.
Yeah, it's core. It's a score coming out of this multiplication.
Yeah, so basically the way you often have to pick this parameter is that you explore values that make the result look reasonable, and then, or you look at the production of virus and you say, okay, I know when my innate response is at the max in whatever units I have, then I know that the infected cells are only producing half as much virus. And so if you knew that at F as its max, P was half of what it was if F wasn't there, that would give you a number for KF. So sometimes you have to sort of back calculate these model parameters based on the things you can measure and you do know. And then you can stick it in there. In our example, I just picked a value that gave me some behavior that looked okay. I mean, this is just sort of abstract here for teaching, but for any real system, you do need to try to figure out, okay, what can I get from the literature to give me values for these? And that's another reason why it's often better to start with a simple model, because the bigger the model, the more parameters you have. And if you have more parameters, you need to basically know your system much better to get numbers for each one of them that make sense. And if you don't have any numbers, then you can make up something, but maybe then it would actually be better to say, well, I just don't know this part, so I'm just going to ignore that in my model for now and focus on the parts that I do know. But that is always a challenge. There's sort of this kind of joke that the experimentalists would like the modelers to include everything in the model, every cytokine, and the modelers want the experimentalists to measure every cytokine and everything, right? And neither one is realistic, right? You can quantify certain things experimentally, and you can put certain things in your model, but at some point it just becomes not doable anymore, or it doesn't make sense anymore. Too complex a model doesn't make sense.
Yeah, because I have been thinking about like using the experiments they are happening in my lab for them. And I I want to go after these variables, like what is the rate that, I don't know, my parasite is growing or dying. But an actual experiment has to be done for me to get it, these numbers. And I can go to literature, but the models for the labs are going to be different as well. Yeah,
I mean, you have three broad options as a modeler. You either start a wet lab and do it yourself. I know some modelers who actually have a mix of modeling and doing their own experiments, or you find a collaborator like Paul who produces data that you can use to model, or you can go to the published literature and see if you can find somewhere there what you need. And sometimes it's a combination. I think most modelers, like myself, have a combination of collaborators that they work with, and then using stuff they find in the published literature to supplement. Any other questions? Okay, then I think we reached the formal lunch break, which might, depending on your time zone, might be fairly late or really early. But anyway, I think we now have an hour break, if I remember correctly. Let me look at the schedule real quick.
Yeah, we'll start back at 2 with chronic viruses, and we'll kind of go right through to about 3:15 or so with the talk and with the questions.
That's if you're in Central Time. If you're in Eastern Time, we're starting at 3. If you're in some other time zone, whatever it is, we start in about an hour.
Yeah, 55 minutes from now. Yeah.
Okay. And again, ask questions in Slack if you have. Keep playing with this, and we'll look at some more later today. All right, I'll see you then.